Hello everyone, in this video we are going to talk about placenta. I know friends, placenta is not a very easy subject to learn, but we will try our best for that you can understand it very well. Now the basic thing you need to understand first, then you should start doing anything. Why we are discussing placenta? What is placenta? Placenta is the thing, is a transporter thing that transports nutrition, gases, and extract products between mother and fetus. We all need this thing. Now, how it will transfer? The function of placenta is not easy. The guy placenta is very tough. He's doing very clever work by filtrating the connection between mother and fetus. For example, if the blood of mother and fetus get mixed, then you know the friends, it's very dangerous for fetus and mother, because the fetus just acts like foreign body for the mother. But still, the communication should be there. The exchange of nutrients and extract products should be there. It cannot be stopped. So the placenta guy is doing very tough work. And that's why we need to understand the anatomy of placenta, how it works. For example, if we see from the early pregnancy, what the placenta has to do? The placenta will go into the endometrium and reach to the maternal blood supply. And maternal blood supply is what? Blood vessel. It's just like a spy infiltrating into another territory and gaining all products or nutrients. So after spying or after infiltrating the blood vessels, again what placenta will do? It will infiltrate and again it will filter the exchange. That means which gas should be passed, which gas should not be passed, which molecule should be passed, which molecule should be not passed between mother and fetus. It will decide by placenta. There are many immunological factors also associated with placenta. Placenta also secretes some hormones also. So placenta is a hell of a creation by the God. So two basic function placenta will do. What? First one is infiltration, like a spying, and the second is filtration. That means selectively choosing which molecule should pass and which molecule should not. So both this function will be there. Now we have already talked about infiltration, how placenta infiltrates into maternal blood vessels. So the placenta have villi. Villi are nothing but the elevated structures. They are made of cytotrophoblast. Cytotrophoblast are nothing but a kind of cells. From cytotrophoblast, another cells are generated, that is called syncytotrophoblast. And this syncytotrophoblast will invade the arterioles of mother. Just like this is an artery, that syncytotrophoblast will infiltrate into arteries also. So now the blood in these arteries can very well pass. So this is what the infiltration. The second thing is filtration or barrier, by which the placenta will act like that. The barrier of the placenta, again, syncytotrophoblast, cytotrophoblast, the blood vessels, the endothelium of blood vessels, all these things will there, so that if the molecule have to pass from mother to fetus, they have to pass through this series of cells, so that the filtration, proper filtration can be done. With this basic information, we will go to how a placenta is formed. My friend, this is a placenta, as you can see in gross picture. This is the umbilical cord. This is the fetal surface. And this is, you can see, is a amnion. This is, my friend, is a maternal surface. So this is a placenta grossly. Now we will see how a placenta is formed. Human placenta is called discoid, hemocoroidal, and decidual. Why these three names are there? Discoid means it is the shape is of disc. Hemocoroidal means this placenta is in contact of maternal blood. Deciduate means there is some maternal tissue also involved. One thing you need to understand, that the placenta made by fetus, not mother. Only some part of mother is involved in making the placenta. Now, let's go how a placenta develops. Development is very, very necessary to learn. Only by this way you can really understand many things, many problems that comes into the pregnancy in further chapters. For example, it has two surfaces, a fetal surface and a maternal surface. Fetal surface is formed by chorion fundosum, and the maternal surface is formed by decidua basalis. And what is decidua basalis? Is the endometrium of pregnant lady. As you can see here, this side is a fetus side, this side is a mother side. These are the villis. Okay, so and the gap between to this villi is intervillous space. Now you can see the villi. We have cytotrophoblast, and above that we have syncytotrophoblast. This line is syncytotrophoblast and these cells are cytotrophoblast, as we have already discussed. The maternal blood vessel will develop between this intervillous space. They are already there, but again they will just regenerate, or they will make branches into this space. Now when the blood vessels are formed, the villis is called secondary villi. This was the primary villi, this is secondary villi. The third thing is, now the fetal blood vessels should also develop, but the fetal blood vessels, the placental blood vessels, develop from which mesoderm? Extra embryonic mesoderm. Extra embryonic mesoderm is the layer from which fetal blood capillaries or the placental capillaries will be formed. So with this extra embryonic mesoderm, when it is added into the placenta, this is called tertiary villi. Tertiary villi is the complete villi. You can see here, this is the cytotrophoblast, above that syncytotrophoblast, this is a blood vessel. These are the fetal blood capillaries formed from extra embryonic mesoderm. This layer here is also a extra embryonic mesoderm. Now we have already told what is placental barrier. That means that if any molecule has to pass from mother to fetus, it has to pass from this different kind of cells one by one. The first cell comes into contact is syncytotrophoblast. From them the cytotrophoblast, then extra embryonic mesoderm, and then endothelium of fetal capillaries. So this, my friend, four structures make the placental barrier. Now if we talk about the placenta, the anatomy of placenta at the term, then it is made of circular disc with 15 to 20 meters, 50 to 20 centimeter of diameter and thickness of 3 centimeter. Average thickness. It is more thick in central area. The average weight is of 500 grams, and this ratio of placenta and fetus should be 1:6. And it covers the 30% area of uterus inside of the uterus. Now if we see the fetal surface, as we have already seen here, the fetal surface made of chorion fundosum. It is shiny gray, covered by shining amnion. Umbilical cord is near its center. You can see here shining umbilical cord here. This is fetal surface. This is fetal surface. Then maternal surface. It is dull red in color, in rough, not shiny. We can see lobes there, 30 to 10 to 38 polygonal areas. One thing that is called cotyledon. What is cotyledon? Cotyledon is a functional unit of placenta, originate from the main stem villus. So the placenta and endometrium. This is, my friend, is uterus, and this is placenta. So this is the placental endometrial junction, and at the junction we have a Nita-Buch membrane. Nita-Buch membrane. Okay, what is membrane? It is nothing but a fibrinoid deposition just above the syncytotrophoblast. So when there is a syncytotrophoblast, below that cytotrophoblast, and here are the blood vessels and all, here is the Nita-Buch membrane above the syncytotrophoblast, between the maternal tissue and syncytotrophoblast. And above the Nita-Buch membrane there is a layer. It is called decidua spongiosum. The separation of the placenta at the time of delivery happens between these two layers, that is decidua spongiosum and Nita-Buch membrane. What is the importance of Nita-Buch membrane? Nita-Buch membrane actually prevents the over invasion of syncytotrophoblast into the maternal tissue. That's why it will be easier to get separated. When the Nita-Buch membrane is not there, then the placenta is morbidly adherent to the uterus, and that will lead to accreta, increta, percreta, very serious complications. So this is the very, very important membrane. Now another thing, fetal fibronectin. It is a trophoblastic glue, and when you see this fetal fibronectin in the discharge, vaginal discharge, it is indicative of preterm labor. Now let's talk about placental circulation. The villi carries fetal blood around 350 ml. Intervillous space is filled of maternal blood 150 ml. They don't mix together because placental barrier is there. Now, what is the uteroplacental circulation and what is the fetoplacental circulation? Uteroplacental circulation established at day 15. And if we see the amount at term, it is 500 to 750 ml per minute, a very, very important and big part of cardiac output. It is 5 liter, 10%, more than 10% of cardiac output. If we see the type of arteries in the intervillous space, they are spiral arterioles. Okay, now fetoplacental circulation, it consists of fetal capillaries. From fetal capillaries, the umbilical vein will be there. The umbilical vein carries the oxygenated blood, and the umbilical artery carries the deoxygenated blood. It is opposite of what happens in normal circulation. Okay, so let's talk about the difference between umbilical vein and artery. Umbilical vein has oxygen saturation of 70 to 80%, while umbilical artery has a saturation of 50 to 60% lesser. SpO2, if we measure, then 30 to 35 to 40 mmHg in case of umbilical vein and 20 to 25 mmHg in case of umbilical artery. What about blood pressure? Blood pressure will be always big in arteries than veins. So here 60 mm blood pressure is there, while in umbilical vein only 10 mm pressure is there. Both of these arteries and vein become a rudimentary structure in the adult life, and the umbilical vein converts into ligament of Teres, and umbilical artery converts into medial umbilical ligament. Now the function of placenta, you know this thing: transfer of gases, nutrients, waste products, secretory products, uh, glucose transportation with facilitated diffusion, fat transportation, immunological factors transportation, various hormone secretion like estrogen, progesterone, HCG, HCl, all these kind of hormones, HPL, sorry, are secreted by this placenta. So this is how a placenta is formed. You need to remember is what? The placental barrier. First comes is the syncytotrophoblast, then comes the cytotrophoblast, then comes the extra embryonic mesoderm, and then there is a fetal capillary which have endothelium. So, and here is the blood, here is the maternal blood, here the maternal blood is there. So from maternal blood, any product has to pass through the fetus. They have to pass through syncytotrophoblast, cytotrophoblast, extraembryonic mesoderm, capillary endothelium, and then they can enter into the fetal capillary. So this is how the placental barrier works. Thank you.